So good afternoon to everybody. It's a pleasure to me for me to introduce you to a new webinar by Affida in cooperation with the Merck Group. It is about the cultivated meat industry and issue related to. So now I will start sharing a few slides. So here we are. This is the webinar of today. Today we have two experts that will help us to understand what about the bottlenecks and the obstacles that we have on the way to a reasonable cost. So diffusion of such kind of a new biotech or biosolution, as some people call, for sustainable one, the contribution of biotechnology to sustainable food for the future. A few words about Affida, that is the organizer, the company that manages this production. We are actually a scientific communication and media company focused on food safety, food quality, and food sustainability. What we do produce, we are editing since a few years ago a magazine that is now even online, of course. We do produce quite a lot of webinars, technical webinars, and we are also organizing international conference. This is the magazine we do make, that is a Affida journal. And here you can see a few webinars we organize. There sometimes are PowerPoint presentation by expert, in other case are panels like today. And then we are organizing also an international conference that is about the what we call science-based solution or actions to make the agri-food system more sustainable. And this is actually the second edition. The first one was in Parma, 2024, and the next one is in May in Venice, Italy. So again, I want to thank Merck Group for the generous support that make this webinar possible for us. So a few words to introduce the topic. I just want to tell you what about is the relevance for now of this kind of production, because it is not only R&D, it's already a reality and a kind of real industrial production or beginning of. Actually, according to the Good Food Institute, there are about 150, that is order of magnitude, it's not easy to map this situation. There are about 150 companies all over the world active in this field. As you can see, there are many in Europe and quite many in Canada and the US, and a few one all over in other countries. Actually, as you know, Israel is very active in this kind of industry, but there are bottlenecks about the regulatory barrier, the regulatory issues, as you know, of course. This is what in Europe we call a novel food. It means that it's something that it was not on the market before. So of course the risk assessment entities, the risk assessment bodies, they want to verify that any kind of novel food is safe for consumer. And so, of course, you need to go to a regulatory approval. And this process is not so slow, relatively fast in comparison to Europe. When we talk about United States, Israel, Singapore, and UK, but in Europe, as you know, in the European Union, as you know, it takes theoretically 9 months, but then it is much, much longer for a number of reasons. They ask the company to write to go deeply into some chapter of the dossier, etc. So in Europe, the process to get the approval is much longer. And here you have an idea how the investment in this area have been implemented. Actually, it looks that in the last couple of years, this was slowing down. Maybe there is a more, the investors are more prudent, the venture capitalists are not so enthusiastic like 5 or 4 years ago, but still they are investing. And if you look at the R&D, the scientific production, considering the scientific publication, you can see that actually the number of publication is constantly growing every year. So approaching what we are going to discuss today, actually we do have two chapters, two group of questions that we would like to submit to our expert. First of all, we have want to understand, me personally, by the way, I'm almost totally ignorant in this area. What about the bottlenecks, technical tech bottlenecks? So what is slowing down or really problematic when you have to grow muscle cells, mainly, not only, in a bioreactor? So what about the issues with the cell line, or any problem with the media and growth factors? What about the kind of bioreactor that maybe are different from the bioreactor that are normally used for producing, I don't know, enzyme by for the or drugs for the pharmaceutical industry? That was this was reality till now in this kind of industry. And what about the problems if we want to create, to produce something that is not just an amount of cells without any shape, but something with a structure? Because at the end, when you go to the market, you will need something that is a little closer to the real meat. And what about the issues related to the need to complement the muscle cells, maybe with some fat and with the aroma, flavor, etc., etc. This is potentially the whole range of questions. We cannot probably get any answer, all the answer, but this is the wish we would have to know, things we want to know. And then there is all the matter related to the challenge to reach at least or approach the so-called price parity, because if a kilo or an amount, a reasonable amount of meat is too expensive, of course, where this kind of industry can go. And again, in this case, according to the literature, the the problem is the cost of media a lot, and particularly growth factors, the amortization of bioreactor, the so-called capex, or the investment for the facilities. Human resources are also a relevant component of the cost, etc. So I found in the literature a couple of papers that could be, if the people want to go more into details of such kind of calculation. It's interesting because in two different papers, on the left side you see one paper of 2021 that is considering the cost per kilogram, possibly in between 37 and $51 per kilogram. And when, according to the other authors, even implementing a big facility and a lot of tons per year, etc., they think that the cost will be more 60 than 50 or 40. So, but we are, even if we think something intermediate, it looks that 40, 50 dollar per kilogram is at the moment what could happen, as it could be the cost. And this is a detail of the second publication, where you can see how the main component of the $64 per kilogram is labor cost, so human resources, the bioreactor, and the grow medium. So now it is the moment to start asking to our expert to help us to understand where the problems are and how we can solve, manage such kind of problems. So we do have now Maria Fernandez from New Harvest, Ramiro Alberio from the University of Nottingham. And so I will kindly ask to Maria and to Ramiro to introduce themselves. Please, the floor is yours, Maria.
Thank you. Thank you very much, Maurizio. I would like to start by thanking you on the invitation for this, for the chance to have this discussion. Do you mind if I share some slides just as an introduction?
Yeah, just a moment. I stop my screen.
And I see if
So
I can share.
if you have anything to share, please.
Yes. Can you see?
Yes, we do. We do. Thank
All
you.
right, so just an introduction to myself. So I have a background in developmental biology and stem cell biology. So as you can imagine, very different from agricultural backgrounds. I entered the cell cultivated field when I started working at Meatable back in 2019. There I did a lot of cell line development. I had the cell line development and tissue engineering department. I was very involved with media optimization, also with the regulatory part. And actually, last year, I've moved from the industry part and I joined New Harvest, which is my current position as Director of Responsible Research and Innovation in Europe. And I would just like to also give a little introduction to who's New Harvest and what do we do. So New Harvest is a global nonprofit organization. We actually exist since 2004. And we're basically trying to build the field of cellular agriculture. So we touch upon a little bit of the topics that you've mentioned, Maurizio. So just as an introduction, also for the discussion, where are we at the moment? What do we see as an organization that deals with not only the industry, but also the academics, with the policymakers, with the regulatory bodies all over the world? So we see that there's quite a big progress in the field. There's a strong B2B ecosystem that it's emerging since when the field started. We see that companies are starting to need pilot facilities to be able to test their processes. We see companies beginning to source raw materials for media production and all of this. We see that institutes for research and training are forming all over the world. Consortia and public-private partnerships are also forming. And we see a lot of policy and regulatory engagements and a lot of supporting on this also coming in. There's also other things that come. There's also things that in the field that still need to be worked on, and we see that early vertically integrated startups are not doing well. So startups that at the beginning propose that will do everything from A to B and they will do all the parts of the development. We see that this is not the model that it's ongoing right now. We saw a lot of lack of pre-competitive research in the field, and I'll talk a little bit about this in some other slides, and a little bit of an imbalance between private and public funding, and a lack of field de-risking. So a lot, besides shared facilities, training programs, all foundational research, that not a lot of data is it out there because a lot of the field was built within these early startups. So one of the things that New Harvest is that the field started as being too private. So against a little bit different than the classic biotech that starts with a lot of public funding. And we move from our early stage research to then product demonstration and scale up. Actually, the cellular agriculture started a little bit the other way around. So there was a lot of private funding, a lot of push for product demonstration and scale up without having this early research being done. And this is where New Harvest actually started, was trying to fund this and trying to bridge this funding gap with the early research and try to bring it through this translational and then to product demonstration. And just to give you, so what we're trying to do as a nonprofit is to support and conduct open research, really to bring this early stage research, breaking down silos between disciplines and sectors, trying to foster collaboration and coordination, and trying to guide a little bit development and governance of the fields of cellular agriculture in general for the public goods. And just a highlight, we're anchored in the US, Canada, in Europe, so you have a little bit of a global view of the fields in that sense. And yeah, and I'll give my word then to Professor Ramiro.
Yep, thanks very much. I will share my slides as well. Where
Yes.
am I? Share.
Very
Thanks very much,
interesting.
Maurizio, for the invitation. And I will briefly say a little bit about, are you able to see my slides?
Yes, we do.
Yes, great. So my name is Ramiro Alberio, and I'm a professor here at the University of Nottingham in the UK. I'm very pleased to be here joining Maria in this webinar. Hopefully, we can answer a lot of questions that are arising in this area. So I'm a developmental biologist. I'm a vet by training, but actually have done research in developmental biology for over 25 years now. And our area of research is stem cell biology. So we are specializing particularly in the production of the cell lines that can be used for cultivated meat. So our lab is based in the UK. We have a spin-out company that commercializes the cells that we produce. So we supply the industry with our cell lines, and we work under funding from a public institutions, so like government funding, but also private. And we also have a lot of collaboration. So in the spirit of what Maria was highlighting, one of the issues in this area is that there was very little collaboration in the past 6 to 7 years. Now there has been a lot more investment in government programs. Karma is one to mention. It's a UK program, a large program that encompasses many universities. Marianne Ellis is the director. We collaborate closely with this hub. And we also have independent collaboration with different institutions. I mentioned Alessandro Bertero here from Italy, which we started the collaboration about a year ago. And we have many others. But just to give you a flavor that the way we work is in a very collaborative environment, because a lot of the basic understanding of this technology, if you like, is lacking at the university sector. So we are working on developing those networks, but we also have a lot of industrial collaborators. Of course, many of the companies we work with are our customers; they buy our cells, but also other collaborators that we have are companies that are within the supply chain that didn't exist until a few years ago. There was no supply chain for cultured meat because there was no cultured meat. Now we are building the supply chain. So we are partnering with different specialist companies that produce either media, for example, like stem cell technologies, or Ocam Biosciences, which makes growth factors. And so we have partnerships for specific bespoke products that we produce and try to provide the industry with a solution. Because the people like IV Farm Technologies, for instance, they make the end product, but they need a very robust supply chain. And this is the area that we're trying to specialize and actually build these networks or collaborations to create the solutions that we need to address the bottlenecks. So this is Pluricells, which offers a platform for acquiring cell lines that we develop in our lab, and it's across 3 different livestock species, as you can see there. Pigs, cow, and sheep is our focus. And these are food compliant cell lines. So these were made with the purpose of making cultured meat. So that means that they tick all the boxes from the regulatory standpoint. And in fact, we are involved in the regulatory environment. So we are advising actually government in terms of what are the requisites for making something food safe. So we are very much interested in understanding what are the compliance issues, and that people that get these lines understand the risks and the hazards that they need to consider when they are initiating this type of products. For those that are not familiar how cultured meat is made, I briefly highlight the 2 main ways of making cultured meat, which is you take either a sample of cells from an animal, in this case a pig, that can come from a biopsy from a different part of the body, and these cells can then be harvested, expanded, and eventually differentiated. Or they can also come from an embryo that has pluripotent cells that can be expanded, induced to differentiate, and triggered to make a product. We specialize on the one that you see on the left-hand side, the embryo side of things. We don't do the adult side, the somatic part. So if you ask me any questions, please direct them towards the left side of the production process, because I don't know anything about the other part.
Yeah, okay.
And in terms of why we specialize on this, because we think this is an avenue that offers hopefully the best avenue for a reliable and reproducible product. I think when you industrialize anything, it needs to be always the same, and that will impact massively cost. So if it's not always the same, there are inefficiencies in the system, and therefore it becomes more expensive or less profitable. So we think that route, and this is an example here of how the different interventions that have taken place over the past decades have led to a reduction in cost. And you alluded to that, Mauricio, in some of the studies. So I have a paper that was estimated about 40 pounds a kilogram of chicken nuggets in 2019. That number is probably gone down by now, but it started on a basis of 250,000 in 2013. So clearly the numbers are becoming smaller and smaller. And a recent work from GFI shows that we should be aiming for a 5 pound kilogram by 2030, according to latest LCA analysis. So we're trying to really, really reduce those costs. How do we do this? There are multiple ways. First, you need to identify the bottlenecks, and this is a kind of a brief outline of the bottleneck area. So from the development of the cell line to the product processing, everything in between needs to be optimized. We have focused on production costs, which is basically reducing the cost of the media, which is one of the major costs. And we're working towards that. To do this, we partner with companies. So the example I'm giving you here is the partnership with stem cell technologies, which we use to produce a new medium for growing our cells, which yielded significant reduction in the usage of the medium, as well as the cost of the medium, which was launched last year and yielded significant increase, as you can see here, in terms of the speed at which the cells grew and the time it takes to have a consistent biomass production. So a reduction of 25 liters or so of medium for a particular batch of cells. So that's contributing to the significant reduction that can be measured and can have a massive impact in large-scale production. Another example of that is the work that we've done with Occam, which is using growth factors derived from plants. In this case, we use a plant-derived FGF. Plants can make this product very easily with tobacco plant in this case, and these are thermostable molecules that can then impact in how much you need to use for the cells, but also how often you need to replace the media. So all examples of how we can little by little and step by step come up with recipes that are not overly expensive and really killing the industry. All the media that we have, initial formulations, were coming from standards used for research or for medicine. So many of the inputs are totally not necessary for food production. And this is the big switch that many of the players in this supply chain are specializing in, trying to produce reagents, products that can be food grade and yield large biomass production. I want to also mention briefly that with our cells, you are able to make a product that you can eat. And I was very fortunate to participate in Meatable's last year tasting session that they had for sausages and meatballs. And I was astonished to see the quality of the product that you could obtain with the right.
And you survived.
And I'm still here. That was actually last year in February. So I was very much privileged to be there, and I really loved the tasting, and I couldn't tell them apart. Basically, what was actually made from a butcher's sausage from the one that they made in the company. So clearly, the product is there. The biology underlying the production of this process is being worked out. We can debate for the next 50 minutes as to what the problems are for the commercialization. I think this is the key of this discussion today, in my opinion, is to identify why can we not scale up? Because the product you can see there, it tastes good, it smells good, it's healthy, and you can eat it. So we can do this. It's just we need to now industrialize this.
Yeah.
It's a new industry starting from scratch. So we need innovation and investment. Anyway, just to finish off, to say that we are very open to collaboration, like many others in this area. Without collaboration, this is not going to go anywhere. The field is growing a lot at all levels, from the end companies that make the meat to the ones that are supplying the different components. And I think there is a lot of scope for further collaborations and consolidation of suppliers in this area that will make this industry thrive. And just to finish, to say thanks for listening. I'm happy to take any questions and contact me if you have any interest after this seminar. Happy to continue the discussion.
Thank you, Ramiro, very much. Thank you, Maria. I think both introductions were very clear and very good for the following debate that I want to start. But before submitting you the question, reminding you the main question, I want to invite the participant, the people that are online, to send us in the Q&A section their question, because we have time also to reply to question by the participant. Please. So we start with what I was listing before. So I would like to dedicate the next 10 minutes, let's say, about before talking about cost. Let's consider the tech issues. So, and first of all, and this is probably the best topic for Ramiro, I suspect, where we are with the stabilization and the reliability of a sufficiently number of good cell line in the, as you explained us, generally you take from an embryo, but are the companies generally still struggling to getting the right cells or not? What they do normally, they are normally mostly developing the line by themselves. There is a reliable market where we are in, which kind of progress we do need just on that side.
Yeah, as I mentioned, so the traditionally, and I'm talking in the past 10 years or so, there were no cell line suppliers as such. So most companies that entered this area had to go and contact academics that had cell lines in the lab based on publications. So there were no purposely derived cell lines for this industry. So there was a lot of back and forth with academics, and also there were some cell lines, primary cell lines that could be acquired from banks, but maybe they didn't fulfill all the criteria that were required for a food grade. So that means that that led to things like what we developed, a platform to actually produce cells that are compatible with food production. And this is in its infancy. There are not many suppliers. There's probably a handful of suppliers globally. There is more than there were 5 years ago, and I'm sure there will be more in the coming years. But it's very important to be to have these strong suppliers of bespoke cell lines so that the industry can start with the best possible material. Because what comes downstream of this, the cell line, is a massive undertaking, technical undertaking. So that means you give yourself the best chance to produce something good at the end and efficiently if you start with the best possible material. And these companies are now becoming very experienced in this. They know the number of hurdles that need to be overcome, from the health of the animals to the derivation processes to the banking system process. So there are quite specialized processes that not companies necessarily involved in the expansion or the manufacturing are necessarily interested in doing. Typically,
Yeah,
people that make a burger don't necessarily kill the animal. They go and buy it from a supplier. So you need to have a supply chain that can be
yeah, supply
specialized
chain.
and knows the detail. And we are now becoming a little bit more sophisticated in this industry, in that we have these suppliers. There is a handful of them, and there is a variety of cell lines that people can go and choose.
And are such cell lines generally stable enough? I mean, I can get the cell line and then going on using it for almost unlimited time, or after a while I do have some problem. Can
I
you?
mean, it depends which cell line. So one can acquire primary cells or immortalized cells
Okay.
or naturally immortal, which are the pluripotent cells. It depends which cell type one chooses. The somatic cells,
Okay.
the ones I showed you on the right-hand side, or the pluripotent cells have different properties. And this is where
Mhm.
you expect the cell specialist, like Maria was doing in Meatable, the specialist in cell line production within the company will know what they actually want. And if
Mhm.
they don't know what they want, they need to get in touch with us, the people like us, and tell us,
Mhm.
okay, what is that you want and what is that you know, and then make those inquiries, because the cell line companies know what to supply you with depending on
Yeah,
what your purpose is.
clear, clear. Maria, do you want to add anything to
Yeah.
that?
No, definitely. Well, I think definitely what Professor Ramiro said is just what there was available 10 years ago is very different than what is available right now, in terms of especially traceability of where these cell lines are coming and characterization. We know much more regarding in terms of regulatory for food safety purposes, what is needed to know. And in terms of stability, definitely there are stable cell lines. The cell lines are very well characterized. It's the starting
Mhm.
point. Of course, then each company will have their own strategy on how they're going to scale up these cells. And from there
Mhm.
on,
Mhm.
there's a whole process of optimization. If there is a good cell line development in each company, they will know how to best produce their cell banks for production for their R&D, and they know how to maintain these cell banks and how to make sure that you have appropriate and fit for purpose for their production that can be kept for years, if not decades, and that are stable within the process that they're developing. But it's about the process development, then that's the trick.
Yeah.
It's about that because you cannot expect that the cell line cells like Ramiro pluricells, these cells they're not previously tested in big batches or in big scales. That's so you also start from a point and you need to understand what's going to happen when you're going to these bigger scale ups. So that's a lot of the technological development that that needs to happen.
Good. So the second question is where we are about the media technology, the availability of, we don't talk now about the cost, but about the quality, let's say, of the media. Are media and growth factors and other materials available at sufficient quality, and we do have sufficient knowledges how to how to choose and use the proper media for getting the right development, the right multiplication of cells, etc. Ramiro, what about?
Similar to the cell lines, there has been a flurry of companies that have dedicated programs of media development specifically for the industry. And again, there is a handful of companies are making media specifically for specific bespoke cell lines. And therefore, these have been thoroughly tested, engineered specifically for these purposes. So one can now, with confidence, go to these companies, get a test batch of media if you want to give it a go first, but they have been validated and thoroughly engineered and designed so that they can comply with a lot of the requirements, particularly with regards to micronutrient, so nutrient utilization. So they don't want to put 2 grams of glucose when you only need 0.5. So it's being optimized so that the usage of the materials in the media is sufficient and there is not a lot of waste, which is typically what happens with conventional media used in cell culture for any of the studies that we do in academia, even for cancer studies or anything. There is just a surplus of everything. Everything is an order of magnitude more than we need. And that's a lot of cost. And so these companies have narrowed down those formulations to the point that they know exactly how much a cell needs at any given time based on the type of bioreactor that you're using.
Yeah, yeah. And what do you think, Maria, about this issue of the media quality?
Well, again, it has to do with which strategy each company is doing because they will adapt cells and it's the holy triangle we want to say between the bioreactor process, the cell, and the media, because the cells, the metabolism, the needs of the cells is going to change depending on which scale we're talking about. And I think that at the beginning of the industry, there was not a lot of flexibility on media formulation. You would get bottled media that you could not adapt as Ramiro was saying, the concentrations to what your cells were actually consuming, and there was a lot of waste. Now, actually, this is one of the things that I mentioned also at the beginning. There's the supply chain evolved with the field. So now there's a lot of companies that are also giving more flexibility or giving powdered formulations, or they're allowing companies to play with the combination of an amino acids. So there's a lot of optimization that it's being done there, and a lot of knowledge that is being created also. And I have to say that I think there's also then, if I want to just a little side of it, when we're talking about cell therapy, for example, I think that there's a lot of learnings that the field of cell therapy could take from what the field of cultivated meat is doing regarding this media optimization and reducing the costs. And I think there's a lot of things that could be parallel. So it's also interesting how this field can contribute to other fields.
And so the third kind of question is about now we enter into the process to how to grow by the way, to a big amount, because in the lab scale is one matter, then you have to scale to bigger and bigger amounts. So the problem is, if I understood correctly, that the experience of the biopharmaceutical industries was with different kinds of bioreactors. I mean, you cannot use the same kind of bioreactor that have been used by the pharmaceutical industries. You need something a little different. So there is a matter even of designing probably step by step the right equipment. So what do you think, Ramiro, about have you had, because you are more on the cells, but probably you have been discussing with some companies which kind of problem they are facing when they do have to commit some company to build for them a custom reactor. They find on the market something that is fitting with their needs or not?
No, and
Uh,
I think we're hitting the major bottleneck of this entire industry.
uh,
Is that the biology is right, the engineering is not.
Mm.
And I think we haven't solved that one. And this is not my area, but I know what the problems are from hearing the frustrations of the companies that have invested heavily in the engineering of the perfect cell. But then you need to make a biomass of tons, and tons is something we've never done for anything else. In pharmaceuticals have dealt always with grams or kilograms, but with metric tons of something is something that's only been done maybe in fermentation products, let's say. So it's extremely, extremely challenging to come up with a solution. I think this is where we need tremendous innovation or rethinking the wheel here. It will need to be completely re-engineered, uh, bottom up, and it's product by design in a sense. So you start really having to understand what you're trying to do at the other end and work out your way because um, I think this is our major challenge, and I defer to Maria. Maybe Maria knows a lot more about this because she dealt with that probably more from her end. But I cannot say a lot more in terms of what the solutions are. I know what the problem is, and this is a serious, serious problem.
Yeah.
So Maria, tell
I
us,
completely
please.
agree with with Ramiro because I think what I've seen, and this is the biology part and the cell biologist part, is we were trying to fit the cell biology and the requirements of the cells into something that already exists, that it's already there. We're talking about these big bioreactors, even on this fermentation scale. We're trying to fit something into that, and it's you can achieve certain levels of productivity on it. But I think that to achieve the levels that need to be achieved, you need to retrofit. You need to do it the other way around, because you're asking a cell that it's quite sensitive to be doing the same as a microbe, and you're trying to really fit and looking at it the same way while we're talking about two different biological entities. And I think we're expecting, and I think this is one of the issues that came with the industry being so, at the beginning, being so privately funded. There was this push to go immediately to commercialization. And the fast way to go to commercialization is to use something that it's already there. And that's why
Yeah.
there was this push to use the bioreactor systems that were available, that there was knowledge about, and then try to push them. I think that that's where you see a lot of the bottlenecks and a lot of why these first generation startups are also getting into financial troubles, because first of all they had to test their process, and they had to build a lot of very expensive facilities to be able to test to bigger scale and put a lot of capex on something that is still under R&D. And that's why I think it's very needed to have these scale up pilot facilities that will allow to be a little bit more asset light and be able to test ideas. But yeah, I don't. I think that the biggest technological development is going to be on the bioreactor side
I see.
and on the production side, and then we add what you also mentioned. Adding to that will come scaffolding, that will probably also generate a specific type of bioreactors and all of that. But then, of course, you get into the supply chain part of it, right? So, but the bioreactors that are used in fermentation are very cheap because you have an industry that, the supply is there already and the demand. Perhaps these new bioreactors that still need to be developed, you still need to create a market for it. Capex costs are going to be very big at the beginning, especially if they're going to be very specialized, right? And then if you think about the industry part, this probably will not drive so much investment, or investment is going to be a little bit more shaky because the return on investment of in this field is not it's still far from.
I see. So who will risk and win? Who will win? Probably now I figure out could be the company or the institution that will develop maybe a completely different kind of bioreactor with different principle, with different design, let's say, in
Yeah.
a way that the cell inside are managed in a different way, because this is the first time that we cultivate 100 or even 1,000 kilograms of mammal cells. That is completely new, right? So
Mhm.
how to manage such a big amount of cells and provide them the nutrient is an enormous challenge. So here, since it's so risky, I think that if we don't have the help by the governments, by the public, it will be an enormous obstacle, if I understand correctly. And this is
You summarize it
connected
well.
to maybe a little connected to the fourth question that is about what we will get from the fermenter at the end. Just a mass of without any shape, or is possible to create some structure, and maybe the cells can grow already on some structure, solid, absorbed on some solid structure. Any research on this side? Maybe we can solve two problems at the same time that we create already in a kind of box. We call it bioreactors, but we can imagine something even different from what we had in the past. A place, a volume where we have structure and a facilitated growth of the cells till a big mass, maybe with some vegetable fibers inside. I don't know, I dream. Ramiro, what do you think about this issue of the scaffolding or structure of the mass of cells?
Yeah, I mean, we need to go back maybe to the beginning. What are you trying to achieve with this industry? The bulk of the companies, the bulk of the technology that is used for this product is not necessarily to replicate what we can obtain from an animal in the sense of a beautiful
Mhm.
steak. I don't think
Mhm.
that's the purpose of the industry as a whole. I don't think. What we're trying to achieve with this is, we have an increasing demand for meat globally. This increasing demand is at around 3 to 4% per year. So we cannot bridge that demand with more animals. However, the most consumed part of the animal comes from the leftovers of the animal because it's meatballs, it's minced meat for making burgers,
Sausages.
etc.
Mhm.
So we as an industry, we have enough problems here that I
Okay.
don't think making a steak is the objective. The
I
objective
see.
here is to reduce the demand for meat for cuts that are totally
Mhm.
useless, like a meatball, a mincemeat, or a sausage. If we can
Mhm.
bridge that gap in the demand,
It's
that will
relevant.
reduce the
Mhm.
demand for animals. So
I
if
see.
we are really clever about what we want to achieve, let's not make life harder for ourselves. There will be someone that will make a printed beef steak because it looks fancy in a Michelin star restaurant. That's not
Mhm.
going to save the world's demand of meat, which is in the metric ton level. So I don't think we have to be deluded by what we want to achieve, in my opinion. It would be the wrong turn to take in this massive challenge. What's the point? However, if you're really thinking about accessibility of this high valuable product, high quality protein, which is in the form of meat that tastes and smells like meat, because I tasted and smells really good,
Mhm.
why not make it in a sustainable way? So if we are very clear about what our objectives are, this is not an unsurmountable problem. I think it's an engineering problem and it can be solved, but we need to be clear that you will obtain biomass that will then combine with mass like plant fibers or other
Mhm.
ingredients to make it a nutritious product that has value for the consumer, and you're not tricking the consumer, you're giving them meat, which is
Mhm.
real. If we are very clear about what we're trying to achieve, I think this is not impossible.
Yeah, I totally agree. Maria, what is your opinion about that?
I definitely agree with that. It's about what is the problem that the industry is trying to solve, and what is also the easiest problem to solve to be able to show that this is a viable way to go. And that starts with this unstructured meat, with minced meat, these type of products. And I do think that there will be what will follow will be indeed requirements for more structured meat. And I do think that there is some technology there. It's already promising. There's a lot of scaffolding also being developed. So we've been talking about cultured meat, but there's also companies and startups that are working on cultured seafoods, so fish, octopus, and those. It's not the type of product that you have minced. Those, it's probably a type of product that if you want to get something out there, you need a more structured product. And they're working a lot in this using scaffolds and testing a lot of scaffolds. I do think that there is a promising, well, as you mentioned, a promising technology out there that involves also a bioreactor type of thing. It's called hollow fiber bioreactor. Merck actually had started a project with that. There's also other companies starting to develop Calvatech and Cellular Agriculture in the UK. They are testing these hollow fiber reactors. So they're trying to solve scalability. So it's a type of bioreactor where you can have very high densities of cells without having a very big bioreactor. And that it, from results and from data that I've seen, it looks very promising in terms of trying to solve scalability and also this 3D thick tissue type of product. So I think there's quite interesting developments in there. Yeah.
And the last question is connected to the previous one. I mean, not about the structure, but about the quality, the flavor, the taste of in comparison. Because, okay, I agree with you, Ramiro, that we don't need to satisfy all the demands for different kind of meat products to be able to create nuggets that are already, by the way, panned, etc. Nobody see or sausages. We can use cells. But at the same time, even if you told me that the taste was good, when you get real meat, we do have an amount of fat inside, for instance. So it's another kind of cell. And connective tissue that give some structure, some feeling even to the consumer, even if it is mashed, if it is grinded. So the question is how we can replicate the quality in that sense. I mean, in the sense of, for instance, fat inside. You have to add a mass of fat cells later, or you can combine in the same box. I say I don't want to call bioreactor or fermenter the cells. And I had an idea, but you use embryo cells. But maybe it is a stupid question, but in the animal, in the embryo, at the end, the cells step by step diversify in a way that they create a muscle. Is it completely impossible to figure out that in the future we, in this kind of fermenter or box, as I call it, we can simulate the process that is happening in the animals? Because you say we go in another way, but and the opposite way would be, can we create a muscle like we have in the embryo, step by step? Is it stupid idea, or is it
No,
can be
no,
possible?
Maurizio, is totally a correct question, and this is pretty much what we most companies are.
Synthetic embryo.
Yeah. I mean, people are doing this. So in regenerative medicine, this has been going on for over 30 years. People have been doing this type of work with human cells to regenerate muscle of injured soldiers or injured patients
Okay.
for donkey's years, like many decades. And we have learned, and also we know the key genes that are necessary to make fat
Okay.
and to make muscle. In fact, Maria can tell you about fat because they've been doing a lot of fat in her previous job. So we make fat, we make muscle, we make connective tissue, the three essential cell types in meat. They can be made to order. And then you mix the proportions. If you want a Wagyu beef, you will have 40% fat or 50% fat versus an Aberdeen Angus, which will have maybe a more lean structure or the composition of that meatball or burger. So you can choose the proportion of cell types that you will make your product for in this kind of way of making meat. And you can even change the type of fat, more saturated or less saturated, to
Yeah,
make,
that is an
so
advantage,
all of
of
this is
course.
possible by engineering. We said before is that the biology is being worked out. It's the engineering that doesn't maybe work out how to scale up. We can make muscle, we can make meat, fat or connective tissue or blood, anything, pretty much anything can be made these days at different levels of efficiency. Uh, from the biology side, the challenge is then to scale up. But I think Maria, you should say a bit more about fat because you worked on that.
Good.
Oh,
Please, Maria.
yeah, no, definitely. So actually, one of the first products that startups in the field, well, the field started with, we're going to make a steak, we're going to make this, we're going to make that, right? And I think that what startups also realize, because it's difficult to scale and costs were not getting down easiest as fast as you would wish, is that what is that people are missing in plant-based? So they also put themselves that question, and there's always this feeling of meatness, juiciness, all of that, and it came, okay, this a lot of this meatness and juiciness and all these characteristics that we associate with eating a steak comes from the fat, actually. So why don't we produce fat and put them and mix with these plant-based products so we can give consumers a little bit a closer taste? So there are several startups that then went into this route. And indeed, fat, it gives, we can produce fat from these embryonic stem cells. You can also produce them from, if you start with other type of cells, but how fast and how efficiently you can produce it, it's what's going to make the difference in the end, of course. And a lot of the organoleptic properties of these meat has been analyzed, actually, at least in my previous company, and it makes a massive difference in how you perceive and how you smell, how you taste. It hits all the notes of it. So definitely not a stupid question at all. That's exactly the question that people pose at the beginning when they start looking at this field.
Yeah. Okay, so this is a good overview of the tech issues. Now we will use another, let's say, 15 minutes about talking about costs, because all such chapters, all such sides are bearing with themselves some cost issues also. So first of all, as I remember from my slide, the apparently the three main chapters, the three main factors are the cost of human resources, that sometimes is maybe the people forget that you have very specialized people at the end, even when it will be an industrial production. You cannot have a simple worker managing a bioreactor. You need molecular biologists at the beginning, cell biologists, and biochemists maybe for the downstream process, I suppose, something like this, and only at the end some food technologists. So it's a complicated team. So this is one of the point. But let's start with the cost of the media, because I think the cell line is probably now not the main problem is to have the right cell line, but Ramiro told us that are available. I suppose this is not the main cost, but I want to start with the cost of the media, where we are with the possibility to, at the beginning it was all the time mentioned as the main problem, the cost of the media. Ramiro, where
Yeah,
we are, did
because
we progress a lot or not?
I think there has been a massive progress on this in the past 5 years, so very rapidly, because people are having those type of studies where they are analyzing the utilization of the different inputs, and they know the metabolism of the cells at the different stages of the differentiation and expansion. So the different phases that the cells has to go through, depending on the origin of the cell, for expansion, differentiation, and then ultimately maturation of the cell. The the optimization of each one of these steps has led to a significant reduction in cost. I mean, in excess of 80% right now. The problem is that we need to now produce a very, very large scale. And so whether the systems to grow these cells are continuous systems or batch systems, that all depends on your bioprocess downstream. So in small scale, one can reduce a lot of the things, but we need to see how to remove the um, byproducts produced by the cells in what kind of reactor system. And then there is even thoughts now or designs to trying to recycle components.
Exactly,
And
exactly. I
so
wanted
all
to
of this
ask
is
that.
ongoing. It's ongoing research, but one can confidently say that the reduction in cost has already been more than, 80% or so of what it was 5, 10 years ago.
Mhm.
But now is where we're starting to think about not only the utilization of inputs, but also the resources. Water is a precious commodity. We cannot use that much water. We need to reduce the use of water, because if we're going to think about massive bioreactor tanks, 200,000 liter tanks, where are we going to get all this water from? And particularly deployed, if this technology is to be deployed around the world, you're not going to expect only 3 companies to produce this in 3 locations. You want to have more deployable technology that can be produced locally, so it doesn't have to travel very fast. It's easy to ship a vial of cells, not to ship 50 tons of meat from um, France to Australia. What's the point? Just you need to manufacture locally if you're thinking of a sustainable production system. So
Yeah.
that means that you need to access to water, for instance. So if you think about the entire, you know, life cycle analysis of your industry, you need to think holistically. And so we need to build a resilient
Yeah, yeah,
method
of
so
course.
that you can tackle everything.
Of course, Maria, what you think also, I'm interested particularly to explore this idea to recycling the same media, because as Ramiro was underlined, if we need all the time again so much water is a disaster. But if we can remove the catabolite and reuse it, we also overcome the problem of a lot of, by the way, sterile water, because even
Mhm.
remake all the time sterile the water is also a cost. Not only you need the water, but you need to filter zero point 0.2, I don't know, or something like this, micron the water, suppose. So
Yeah.
it's a big problem. What
Yeah.
do you think, Maria?
Yeah. No, well, Ramiro definitely hit all the points. In the end, it's about how your cells are using your media, right? So you want your cells to use your media the most efficient way. And nowadays, the cost media is also a specific component from the media that are the growth factors that are usually the drivers of these costs. So growth factors are molecules that guide the cells to grow or to differentiate, but that the guide their homeostasis. Um, it's about efficiently using your media. So it's not you can have a cheap media, but if you're going to use a lot of media to get a certain amount of biomass, in the end, your production costs are high, right? So independently of the cost of the media, it's about what is all your developmental process to use a cheap media that you use a little bit, because usually the difficulties is it's finding a balance between it, because if you give cells an expensive or a relatively expensive media with everything that they need in a calm environment, you're going to get a certain productivity out of your production. But then you need to reduce costs. If you start reducing costs, if your media starts becoming cheaper and cheaper, the cells might not grow as well as they were growing at the beginning. So in the end,
And
you're losing productivity. You can have a cheap media, but then your cost is higher because you need more input media. So it's a balance on how much you want to get your media to be cheap, but getting the same production in the end. I do think that that's not going to be the bottleneck. In the end, we've seen really dramatic decrease in costs, even amino acid in all this, with all the supply chain being built around it. I don't think that's going to be the difficult part. I think there's some R&D that still needs to be done on understanding fully the metabolism of the cells and what keeps the cells happy in the bioreactors that are being used nowadays, because we're trying to force these cells to grow in these, as we discussed previously, it's trying to force these cells growing in very big bioreactors. There's a lot of shear stress, a lot of forces in there. We might be able to get an even cheaper media if you put the cells in a little bit of a nicer environment with a different setting with a different shape or a different standard of bioreactors. So in the end, you cannot separate these 3 parts. They all influence each
maybe we should create another kind of reactor, like a synthetic intestine, where you produce the blood, let's say, that is the
other.
media for the body, in a different step, not
Or
to
in a way
buy
of,
the
yeah,
media, not to
or in
buy
a
the
way of
media.
also filtrating ammonia, lactate, these type of things that in the end can influence cell growth. But there's in the end, you want to have a simple process, right? Because the more you put in the middle of your process, the risky it is. You're going to have more,
Exactly.
you have more points of attention.
Yes.
You're going to have more contaminate, as you mentioned, more issues of possible contamination points. If you need to have a lot of process steps in between, the risk of failure is big. So again,
I
I
see.
think that the process that will win was going to be a little bit a balance between all of these points. And there's a lot of development to be done there still.
Now we go to the step that before, when we were talking about the tech issues, was identified as the most critical one, that is the availability of proper bioreactors or innovative bioreactors. I see that that point is the real main bottleneck. And so connected to that, there is a problem of cost of such. Even the traditional bioreactor are quite expensive, but if we have to re-engineer the equipment, that we need an R&D before the production of the just purchasing of the bioreactor. So Ramiro, this is still probably or even a bigger problem for on the economical side, isn't it?
Yes, and I think the challenge is to think which companies, what kind of industries today are do have that kind of infrastructure potentially to redirect it to this type of application, and it's very difficult to find such things. There are meat manufacturers out there that have interesting capabilities, but really they're not bridging some of the essential aspects of the expansion of the cells in a whichever form we want to package them that really accommodates our needs. So I think there is a need for innovation here to come up with a new
Yeah.
way to do this. That I don't think exists yet. I haven't seen it. I mean, there are interesting prototypes. There are many ideas,
Uh-huh.
but I think they're still hitting the bottleneck of scale. Like we're talking massive scale. We're talking here hundreds of thousands of liters or kilograms of meat.
Yeah,
So,
yeah, yeah,
yeah, maybe a couple of hundred of this and that is fine, but that's not commercially viable. We are trying to replace something at a scale that has never been done for anything before. Nothing has been made at this
yeah,
scale.
yeah.
So it's something quite, it will have to require a step change in the thinking from
Yes.
engineers to come up with something that is viable
Thi-
in a biological system.
This is not a job for the startups that are trying to develop the meat. This is a job for the engineering company. Maria, what do you think about?
Well, I think it's a job, it has to come together, right? Because the
Mm,
engineers also need the inputs of,
Yeah,
they also
they
need to
don't understand
understand the
biology.
biology of it. Otherwise, they're developing something for
yeah,
nothing in the end, right? I do think that there's has to be some more incentivization, sorry, difficult word.
incentives, yeah.
Incentive, thank you, for so the startups on the engineering part, right? And that there has to be more funding and more money being put into having some prototypes out there. You don't have this. If you're going to see the pilots, the scale pilot scale factories that are out there, they are dependent and limited by what is commercially available out there.
Yeah.
So companies can come and go, okay, I want to develop a process, but in the end, my process needs to fit this, needs to fit a box already. I really would like to try something completely different. I
Yeah.
would like to build a completely different bioreactor. That's not there yet. You don't see that because
Any European European founded R&D project in this area, up to your knowledge, Maria? Because the EU should take this responsibility. If we want to have an European industry successful in this area, that is strategic or long term,
yeah.
because I don't believe the cultivated meat is something that will be on the market in 5 to 10 years, maybe 10 to 20 years. But
Mm.
at the end, I believe, like Ramiro was underlying, that is one big contribution to the need of meat, because
Yeah.
and engineered meat, by the way, with better nutritional profile than
Mhm.
the
Mhm.
normal meat is
Mm,
ideal. So
mm,
it's so I think it would be reasonable that there will be specifically on the bioreactor issue some EU-founded research project where
Yeah.
engineer and
Yeah.
biomolecular cell biologists design together something. They try something even completely different, because
Yeah.
this is TRL 4 or 5 or even less, as
Like,
we
mm,
say
yeah.
in the language of
Yeah.
the project. We cannot rush to TRL 8 or 7 when we are not there. It's
Yeah.
crazy. It's wasting of money.
Yeah. So actually, it's funny that you're mentioning that there's a couple of EU Horizon projects that we are involved
Okay.
with. One of them being Feast, the other one is called Epic Shift. Feast is more directed to cultivated meats. What they're doing is really doing a technical assessment of what are the methods out there, what is the state of the art on this, and they're also trying to come up with a recommendation for the European, exactly what you meant, and to try to come up with a recommendation based on data that is there or newly generated data within the consortium. Recommendation for the European Union on for this to really become a thing, or for this to be able to, we can pick up the fruits of this in 10, 20 years. This is what needs to be done right now. So this is
Wow.
this is happening in these projects. Um, and yeah, ideally the European Commission or there would be a will of investing in technologies that are, as you mentioned, TLR 4 or 5. But as you know, also coming from Italy, this is it's not an easy topic, right? Let's not go that way, but it's an
Oh, I'm
easy
not from
topic.
Italy, where I am. Because
Um,
actually, it's not only Italy, even Florida and the other US state
it
decided to
become a
stop.
political it became a political
Yeah, that is.
argument as well. But it is important that we also forget that this should happen. We should take the consumer and all the other players in the field. We should take them into consideration, right? Because indeed, it's not only about bringing the cultivated meat. It's about the impact that will have in society, the impact that will have in our traditional agriculture, and we cannot just ignore that they exist. We need to bring them along and also make sure that, you know, the impact is it's taken into consideration and understood. So
So think good. We have a few minutes more, and we have a few questions, by the way, from the audience because we already were scanning almost everything, I think.
Mhm.
But before going to the end, I will read a few questions that we got. One question is, first of all, saying he wanted to he wanted to thank us because of the really interesting and engaging discussion and presentation. I'm interested to hear what the panel's view on any measurement need and challenges for analytical traceability of cultivated meats or indeed products as a result of cellular agriculture in the food and feed supply chain. So the issue is how to guarantee the traceability. Ramiro, what is your opinion about this question?
Yeah, I know it's very important for consumer confidence. We need to make sure that what it says on the tin is what's in the tin. So, absolutely, and this will be a regulatory requirement. So the regulator will definitely require a set of guidance, as it does for conventional meat products, by the way. You need to demonstrate that if you say you have chicken nuggets, that there is chicken inside. So it will be
Mhm.
exactly the same for this. So analytical and traceability of the health status of the cells that were obtained for this process, as well as what comes at the other end of the bioprocess that hasn't been mixed up in the middle. So all this will have to be demonstrated with a it will be
Mhm.
on the manufacturer to demonstrate that purity of that product, of course. Like any other food product, this is treated exactly the same.
Yeah, I understand. Probably by PCR, I suppose.
There are many ways to do that, but PCR is
Molecular.
one easy one but also traceability. If you say you're selling a bird in Angus, is it?
Mhm.
I mean, so you need to demonstrate that beyond PCR. There will be um, other assays that will have
Other
to be
methods.
used for
Mhm.
for this.
Maria, do you have anything to add to that?
it's just yeah, labeling is going to be very important, right?
Yeah.
Because we also want to make sure that the consumer knows what they're buying, of course.
Mhm.
And it's not only about traceability of the product that you're selling, what it is, what you're selling, but also then let's talk about food security or microbiology security, that there's no
Yeah, okay.
risks of foodborne disease and things like that. But those methods are not going to be much different than the ones that are already existent. So I think that
Yeah.
there's a lot of methodology that can
This
be adapted
the
already
sec
for this.
the second, sorry, but we have a limited time.
Mhm.
The second question is 2D data looks promising. Will this cell stable in suspension culture? If yes, will it be stable at least 50 to 100 doublings to have consistent productivity? If no, how could we make it stable at least 75 to 100 doublings? Ramiro, this is a
Yeah, of course.
question.
I mean, I just briefly showed 2D data just to entice the audience. We have work on 3D now and look very promising. And again, it's much of the way we're doing is adapted from what has been done with human cells for therapy. And our cells are very happy in this 3D condition. So
I see.
100 doublings is actually nothing. It has to be way more than that to become
Yeah,
actually.
yeah,
So we are
I
working
suspect.
in something a much bigger scales than the 100 doublings. And this is technology development. It's going on. There are challenges, but it looks extremely promising because the challenges are a bit different to 2D in that you need to decide what time you decide to split the growing clumps, the cells grow in 3D clumps. And there is a need to adapt again the scalability of this into continuous cultures. And so you're adding additional layers of complexity to your reactor when you go in
Mm.
3D. The point to say is just it works. Now it's about, again, engineering the scale of that work. The cell can grow in suspension, and that's a very important, from the biology side, a very, very important starting point.
How to have enough happy cells, Maria, in 2D or 3D? What do you think about?
Oh, it's really about understanding your system and understanding your cells and having a constant, you need to really keep an eye on them. Let's put it that way. And really, it's about how you control your production process and how you control
We have a
your
third question. Should we expect the systematic use of preservatives for laboratory meat preparation? This is not the case for conventional meat from slaughtered animals. So what about the need for preservatives, Ramiro?
I mean, I anticipate the laboratory made meat is meat. So you should treat it the same as meat from the preservation point of view. It's a muscle cell that will undergo the process that a muscle cell undergoes when you kill an animal, the rigor mortis, and so you assume that you will undergo similar process of protein degradation as a conventional fat
Uh-huh.
cell or muscle cell. So I
Uh-huh.
don't see why it should be treated any different. But again, I'm
Uh-huh.
not sure meatballs are treated the same as a steak. So there might be a level of preservative added to a meatball or a burger that is not added to. I'm not familiar with what conventional meat production looks like, but we have to start from the assumption that this is meat. It's not
Yeah,
different to
yeah,
conventional meat, so it should be
yeah,
treated
yeah.
at least the same, maybe.
I see, I see. Maria, have you had any discussion about this side with companies, with colleagues?
Well, so it depends again what the final product is. I could again what Ramiro is mentioning, if it's a meatball or if you're doing this hybrid product that you're mixing it with plant-based,
Mm.
there it might be, but that's on the product development part, that's more on the food
Yeah.
technology. In the end,
Mhm,
what's your
mhm.
product? During the process itself, also preservatives and things like that are not expected to be used.
Neither preservative nor antibiotics to avoid
No.
contamination by bacteria. No, you don't need that.
You don't add antibiotics to the meat that goes from the abattoir to the slaughterhouse to the supermarket,
No,
right?
I
So
mean in the fir-
During the process.
in the
no. Well, I
Yeah,
biofermenting.
that's
mean, no. I mean
No,
that's maybe we haven't talked about antibiotics. We could have talked a whole hour about it. Antibiotics are maybe only used at the very, very beginning of the isolation process because it's
I
inevitable.
see.
But that's a one-off, and it's I don't know, within a week it's gone. All the stuff that is the downstream bioprocessing should be done antibiotic free if it's a good facility.
yeah.
Yeah, it must be
I
food
mean,
food
yeah,
grade and even
yeah, it should be sterile.
Yeah.
sterile. Exactly, exactly. It's definitely much better than a slaughterhouse, by the way.
Oh, absolutely. That's the
Cleaner.
problem. Yeah, it's too clean. Too
Actually,
clean.
this is we have to underline that we have a number of problems, safety issues with the meat, with the traditional. Because all
Hmm.
the time we talk
Hmm.
about other aspects, sustainability and
Mhm.
this and that, but we have to consider how many coli
Yeah, contaminated cases. Yeah.
proper
Hmm.
contamination and infection we do have,
Yeah.
even toxic E. coli we have in the normal meat food chain.
Mm.
Yeah.
And this, if we manage this kind of production, we can be totally free from that
Yeah.
because this come from the intestine of the animal, 90%,
Yeah.
And the slaughterhouse cannot be sterile place. This is completely
Sure.
far from. Okay, guys, I think we have had, now is already about 1 hour and 15 minutes. I think it was quite interesting. I thank you very, very much, Ramiro and Maria. I hope the participants were satisfied. We will send them an email to ask, like a kind of poll, if they consider that it was satisfactory. I think should be, definitely. I thank again Merck Group for sponsoring this webinar. And maybe we can in the future explore other side or go deeply in with other companies, with other experts in what we identified actually as the main bottlenecks, that is both technical and probably regarding the investment involved, the development of real innovative bioreactor. This is my conclusion, because all of the other, on the other side, we are progressing a lot, but still identify a big problem, a big fragile point in the facilities, and particularly in the bioreactor, that maybe we have over there to invent something completely new. So we need on that step to go back to almost fundamental or early stage of R&D to create something that is really innovative, completely probably different from what we have been using in the pharmaceutical industry and in the traditional fermentation for food. Thank you very much again.
Thank you very much for your time. It was great fun. And thanks, Maria, for sharing this panel. It was good.
Thank you. It was great. Thank you so much.
Thank you very much. Have a nice
Bye-bye.
time.
See you.
Bye-bye.
Bye-bye.
Thank you to everybody.